Recording. Okay, so we have two immunology presentations. This one's a little shorter, so we'll start with this. This is a cytokines breakdown. If you remember, we did like a physio lecture last week where we introed the different cell types, talked about granulocytes, talked about lymphocytes. We're talking specifically about cytokines in this lecture, and then we'll do one more that kind of wraps up the rest of the immune system, ties in some immune path, and even kind of revisits a couple genetics concepts. So it kind of pulls a lot together that we've gone through so far. But this is like the last portion of the immune system I would really focus on would be your cytokines and or your chemokines. So what's cytokines, just to remind you, they're signals that are generated by binding of a ligand, something to its receptor, right? An antigen to its receptor. Cytokine receptor binding is non-covalent. It typically is high affinity, meaning it's a strong bond that's hard to break. It typically will result in a transcriptional change of the target cell. Typically, it's going to tell the cell to increase or to decrease either other cytokines or other cell types. And a cytokine signal is any event that instructs a cell to change its metabolic state. Cytokines are proteins. They can act as endocrines, so they can act in the blood. They can act as paracrines released to affect nearby cells, so they can act as autocrines to actually essentially affect the cells that release them. So they can act in all three ways. So some general properties that it's good to just kind of know these terms or at least have some name recognition of these terms. So pleiotrophic activity means that there's going to be a different biological effect of the effects dependent on the target cell. Redundant means that essentially it's going to be the same effect on the target cell. That makes sense. Redundancy, same effect. Synergy would be the effect of two cytokine activities together is going to be additive. We see this often with inflammatory cytokines. If you have one inflammatory cytokine and another, and you add them both together and they affect the same cell, it's going to increase that pro-inflammatory effect. You can also have antagonistic effects where one cytokine's effect inhibits the other. You can see this with pro-inflammatory and anti-inflammatory cytokines where they can essentially block each other's activity. Or a cascade effect where one cytokine on one target cell causes that cell to produce an additional cytokine, almost like a domino effect. We're going to go through classes next. So when I talk about the classes, this is where things can get confusing. So there's 6 cytokine families that are created based off of the structure of the cytokine itself. And so you'll see here that there's some that are really helpful, like interleukin 1 has its own family. So that's great. IL-1 we'll talk about on its own. But class 1 has pretty much all the other interleukins in it. So that's not super helpful. So we're going to break down class 1 further, besides just its own class. Class 2 is all of your interferons for the most part. Then we have our tumor necrosis factor family, which is going to be our tumor necrosis factors. We have interleukin 7 on its own, and then we have chemokines. So we're going to break down kind of some of the functions of each of these, but we will break down like this class further into each of the interleukins to talk about what their specific functions are and who secretes them. Now I'll kind of present it in a few different ways that you could use to memorize, whatever way is helpful for you. So if memorizing by class and functions helpful, great. Memorizing by who secretes what, I have that list too. Awesome. Or we could even memorize, there are some mnemonics that I'll throw out there as well. But we're going to start with our interleukin 1 family, our IL-1. It was our first non-interferon cytokine that was identified. And these are typically more inflammatory, pro-inflammatory mediators. So these promote inflammation. You can have them released by a virus, a bacteria, or a parasite. They're secreted typically early on, and they're secreted by macrophages and dendritic cells. They're going to act locally. So this would be paracrine action on capillary permeability to essentially allow for the movement of leukocytes, white blood cells, to the tissues that are infected. They also could act systemically via endocrine action to signal the liver to produce acute phase proteins. And they can also help initiate our adaptive immune response. So that's our big IL-1 category or family. So know that they're pro-inflammatory. They are secreted in response to any number of the infections. They are secreted by macrophages and dendritic cells. I think those would be the 3 things I would really keep in mind for IL-1 to start with. When we look at class 1, this is where we will break it down further. So class 1 cytokines as a class They're very diverse in action. That tells us nothing. They are a single protein family. That's why they're in the same class together, because they all have the same kind of protein backbone structure, but they have multiple subunits. So now we're going to get into kind of breaking down cytokines that you should know based off of who secretes them. I didn't really have a better place to put it, so I just stuck it here. So the cytokines secreted by macrophages in the acute phase are going to be IL-1, IL-6, and TNF alpha. Then afterwards, they will recruit IL-8 and IL-12. So these are all secreted by macrophages. So 1, 6, TNF alpha, 8, and 12. 1, 6, and TNF alpha come first in acute inflammation. Our T cells will secrete 2 and 3. So I think about T cells, there's 2 types of T helper cells, maybe, or 3, you could say 3 types: T helper, Th1, Th2, Treg, however you want to remember that. But T cells secrete 2 and 3. Th1 cells specifically secrete interferon gamma, IFN gamma. Cytokine secreted from Th2 is 4, 5, 10, and 13. Come up with some mnemonic for that. I don't have one, sorry. And how to remember the interleukins and function. This is a mnemonic. It's your hot T-bone steak. I don't know if you've heard this one before, but fever is hot. That's IL-1. 2 is stimulates T cells. That's your T in T-bone. Stimulates bone marrow. That's your actual bone in T-bone. That's 3. IL-4 stimulates IgE production. That's your E in steak. IL-5 stimulates IgA production. That's your A in steak. And 6 stimulates acute, with a K, phase protein production. That's your K in steak. So IL-1 through 6 are generalized functions, or who they stimulate. You could remember via hot T-bone steak, the HTB, and then the EAK. And then that's one cytokines, essentially interleukin 1 through 6. A little bit more on IL-1, since we just finished that. It also can be called osteoclast activating factor. So it can activate osteoclasts then and cause some bone breakdown. It causes fever and acute inflammation. It can activate the endothelium to express adhesion molecules. It can induce chemokine secretion. That just is essentially what I said before, pulling white blood cells to the area of infection. And it can do that via vasodilation, so that white blood cells can actually move in and out of the blood cell or the blood more effectively. That's your IL-1. IL-2, remember it's the T in T-bone. It's secreted by T cells. It stimulates the growth of helper and cytotoxic T cells. And it also stimulates the growth of regulatory T cells. So it's helping all of our T cells grow. So it's secreted by T cells, and it activates or induces stuff in T cells. So you could think of that as autocrine action. It also stimulates the growth of natural killer cells. That could be paracrine or endocrine action, depending on how far away they are. IL-3, remember this is the bone in hot T-bone steaks. It's secreted by T cells, but supports growth and differentiation of bone marrow stem cells. So bone marrow stem cells, so we're thinking about production of all kinds of cell types, including B cells, things that are developed in the bone marrow. IL-4, that's going to be the E in steak, secreted from our Th2 cells. So it's going to cause IgE, right? It's going to help with stimulating IgE secretion, the antibody. Th2, all responsible for the allergic or parasitic response. It'll induce differentiation of T cells in the T helper 2 cells. It's going to encourage the body to create more Th2 cells. And then it will also promote the growth of B cells, and it'll enhance class switching to IgE as well as IgG, but you'll get a lot more IgE, that allergy or parasitic response. You can think about this as ain't too proud to beg for help. And that's the things that are essentially underlined here. To beg for IL-4 help. IL-5 are A in steak, secreted from Th2 cells, promotes growth and differentiation of B cells. It'll class switch your immunoglobulins to IgA, your mucosal antibody. And it'll also stimulate growth and differentiation of eosinophils as well. Th2, our allergy, and our parasitic response. I5 base, so you can remember that one. IL-6, it'll cause fever, stimulates production of acute phase proteins. Again, these are all acute inflammatories. secreted by macrophages. IL-6, we talked about that one at the beginning. IL-8, major chemokine factor for recruitment of neutrophils. Chemokine, think about it like almost like a pheromone, right? Something that a neutrophil could smell in the body via chemicals and be attracted towards. It's going to essentially tell neutrophils to travel towards that chemokine or that chemical signal. Macrophages secrete it. Clean up on IL-8. IL-8, interleukin 8, is going to be your major neutrophil recruiter. IL-10, Th2 cells secrete. It's going to attenuate the inflammatory response. It could potentially calm it or balance it out. It'll decrease the expression of MHC class 2 as well as Th1 cytokines. So this is a helpful balancer, right? MHC class 2 is going to bind to our CD8 cells, our T helper cells. It'll inhibit activated macrophages. It'll inhibit activated dendritic cells. It also can be secreted by Treg cells, which makes sense if it's regulating the immune system. Tumor necrosis factor beta and IL-10 both attenuate our immune response. So these could be the calmers or the anti-inflammatory cytokines that you should know. They help calm the immune system. They help balance the activation of the immune system. IL-12 helps differentiate T cells into Th1 cells. So instead of Th2, we're getting more Th1, more intracellular cell fighters. They'll also activate our natural killer cells. It'll facilitate a granuloma formation in tuberculosis, so helping kind of wall off that bacteria from spreading, which is not great for having us treat it, but is good in preventing it spreading to other tissue. It's secreted specifically by macrophages. IL-13, secreted from Th2 cells, promotes IgE by B cells, induces alternative macrophage activation. You could think of interleukin 13 promotes IgE, all the E's. That was like all of our class 1s, right? So we had interleukin 1 and all of our class 1 interleukin cytokines. I would focus on just knowing something about each of them. If you're able to know who secretes them and in general something that they do, that would be great. That'd be awesome. If you at least know that IL-10 and tumor necrosis factor beta do the opposite, right? They're not inflammatory, they're anti-inflammatory. That'd be great. If you at least know that, that would be great. If you can remember hot T-bone steak, cool. If you can remember like a next step, it would be knowing one thing about each of them in addition. Our class 2, our interferon class, we've talked a little bit about them, but we'll look at them a little closer. These are all going to be IFNs, interferons. There's technically 3 types. There's interferon alpha and beta. They typically have a lot of antiviral properties. So think about them when you're dealing with a viral infection. They are secreted by macrophages and dendritic cells. They're going to inhibit protein synthesis, and they're going to induce synthesis of ribonucleases. So if we think about viruses, viruses try to hijack a cell's nuclear DNA machinery to replicate themselves. So interferons then are going to stop replication of that cell type to prevent viral replication. So that's actually a helpful process. So we're inhibiting our own cell's replication process to prevent a virus from replicating and hijacking the system. So if we look at our function of interferons, they are involved in our innate immune system. They interfere with both RNA and DNA viruses. And a cell infected with a virus will synthesize these glycoproteins. That's what an interferon is, a sugar protein. They will act locally on cells that have been essentially tagged at being infected, and they will prime them for a viral defense by down-regulating protein synthesis to resist viral replication. They'll also upregulate those cells' MHC expression so that they're tagged by the immune system as cells that are infected, so they can either be treated or destroyed if they're essentially past the point of salvage, which most of them would. You also could see interferons involved in anti-tumor activity for that very reason. If you have a cell that's cancerous, that's growing a tumor, or it's growing cancerous cells, or it's in a dysplastic or a metaplastic state, you could see the same functionality of your interferons, targeting, stopping replication, tagging it with more MHC proteins so they're essentially recognized by the immune system and eliminated. Okay. Interferon gamma is a dimer produced by activated T cells or natural killer cells, and this is involved with adaptive immunity. Interferon type 3, I see this one discussed probably the least. It's involved with viral replication as well and host cell proliferation. But I would say alpha, beta, and gamma would be the 3 that I would focus on the most. Alpha and beta being involved in that viral defense system, gamma being part of the adaptive immune system, helping to modulate adaptive immunity. More on this, so natural killer cells and T cells release it in response to antigens or even in response to IL-12s. This is part of that cascading effect, that domino effect. It'll tell macrophages to kill phagocytosed pathogens. So if they have interferon gamma, the macrophage will then know, okay, this phagocytosed pathogen that I have now identified, I now need to go and anytime I see this tagged on a cell, I need to phagocytose that cell and essentially eliminate or wipe out that cell type. It'll also inhibit the differentiation of Th2 cells. It essentially will promote Th1 response, which is our intracellular response, allowing the body to target things that are inside cells more. And it will induce IgG antibodies, so IgG class switching, so from IgM to IgG, again, further promoting intracellular destruction. MHC expression and antigen presentation by all cells will be increased with interferon gamma release. Okay. So that was our class 2. Now we'll look at tumor necrosis family. We already looked at tumor necrosis factor beta, but we'll look a little bit further at some of the other family members. These as a whole, the tumor necrosis factors are regulating development, effector function, homeostasis of cells, especially of the skeletal, the nervous system, and the immune system. They could be soluble or they could be bound to a membrane. So they could be floating throughout like circulation or bound. This is talking more about what they look like. I don't care about that for the purposes of this test. Tnf alpha is pro-inflammatory. It's produced by macrophages. Tnf beta is produced by lymphocytes, and it will deliver signals to leukocytes and endothelial cells. Remember, Tnf beta is one of our what types of cytokines? Attenuating, right? Calming for the immune system. So you can think of tumor necrosis factor alpha as pro-inflammatory and tumor necrosis factor beta as anti-inflammatory. That'd be okay, an okay, appropriate association to have. There's many bound TNF family members. I wouldn't worry about your bound members of this family for the purposes of this test. Just know they do exist. I would pay attention to TNF alpha and beta. Those are the 2 that I would pay attention to. More on TNF alpha. It's secreted by our macrophages. It activates endothelium, meaning that it's going to allow endothelium to let cells move out of the endothelial space into tissues. It can help with recruitment of more white blood cells. Vascular leak, I already mentioned that, can help increase fever and sepsis. It works along with IL-1 and IL-6 for that kind of fever response. Pathologically, it can cause cachexia and malignancy, and it helps to maintain a granuloma in TB. So when it goes wrong, right, it can help essentially something that's walled off, stay walled off. Okay, that's our TNF factor family. IL-17, this is the most recently discovered. You'll see that they are pro-inflammatory. They promote neutrophil accumulation and activation. They're expressed on a variety of cells. The receptors are found primarily on neutrophils. They also can be found in keratinocytes or other non-lymphoid cells, but neutrophils are like big actions from IL-17. They will work at the communication point between innate and adaptive immunity. So kind of that point that between the 2 systems. All of them are transmembrane proteins. So they are actually located in an actual membrane. They're not secreted into systemic circulation. There's lots of different subtypes. Do not know them. Just know that they're pro-inflammatory, and neutrophil recruitment is involved in almost all of them. And then lastly, our chemokines. These are trying to attract things to a location. They're chemoattractants. So they're going to help affect direct leukocyte migration, meaning they're going to try to tell white blood cells and cells of the immune system to go somewhere. Disulfide bonds or sulfur is typically one of the hallmarks of their protein makeup. And they also will have like some kind of shared residues. You don't need to know that for the purposes of this test. They are, all their receptors are G protein coupled receptors for the most part. So they're acting through that GTP process where we have the G protein, activation of cyclic AMP, so on and so forth. So when a chemokine binds to its receptor, it's going to have some second messenger property using a G protein for the most part. Majority of the receptors can bind to multiple chemokines, so that's why they can have a lot of different activity. And it will just help cells move from one area to the other. This is just kind of talking about the process as a whole of what antigen signaling is including. And that is that. Okay, we just took a break, so I'm gonna, we'll maybe take a 5 to talk, where if you have any questions, I'm happy to answer them about chemokines and cytokines, what you should know. If you know one thing about each, that's like already well beyond what you need for the test most of the time. They'll always have 1 or 2 questions that are more specific than what you've probably learned, and that's fine. There'll be at least 1. For the most part, they'll ask who's secreting or who it's trying to recruit, or if it's pro or anti-inflammatory. Yeah.
Past tests, have you seen Tnf Beta referred to as the lymphotoxin A?
Never. Yeah, I always seen it as Tnf Beta. Yeah, good question. I included there just so you have that, but I've never seen it referred to on an NPLEX exam. Definitely on a USMLE test, but not on an NPLEX. Great. All right. Do you want to break or do you want to just power through? We have an hour. It's up to you. Power? Let's do it. All right. I'm going to stop the recording just so I can have a new. So we'll